In this video, we will see the steps of meiosis in an animated view. This is a long thread-like structure called as chromatin fiber. Here for representation, we will show the two chromatin fibers that represent two chromosomes. Next phase called S phase, in which the DNA of each chromatin fiber get duplicated so that the chromosome number remains same but DNA gets doubled. Also the centrosome get duplicated. In the lactotine, the chromatin fibers now they are condensed to form the chromosome structure. In the zygotine, homologous pairing takes place. This involves a protein complex called synaptonymal complex. Here you can see one bivalent having two chromosomes with 4 chromatids. Number of bivalent can be calculated by dividing the diploid number to half. In the pachytene stage, the next stage after zygotine, the non-sister chromatid exchange their fragment by the process called crossing over. In the next stage, diploidine stage, the synaptonymal complex dissolve and the crossing over point called chiasmata is visible. The next stage is the called dikinesis, which is represented by the movement of chiasma toward the terminal end, that is called terminalization. And at the end, the nuclear envelope disappears, astral rays begins to form, and the chromosomes are attached to the spindle fibers. Each chromosome is attached to one side by a single spindle fibers. They are then brought to the equator, forming double metaphasic plates. The next stage marks the movement of two chromosomes toward opposite poles. This is called Anaphase 1. They reaches to the opposite poles. At each poles, the nuclear envelope reappears, forming two daughter nuclei, and cytokinesis simultaneously takes place, forming two dyad cells with half chromosome number. Next is the interkinesis stage, where only the centrosome duplicates, and then meiosis 2 begins. In the prophase 2 stage, the envelope disappears, two astral rays forms, and they bring the chromosome at the equator. This is the metaphase stage. In the anaphase, the two chromatids get separated from each other due to shortening of spindle fibers. Here these phases are exactly similar to mitosis, but here important one is that they all show variation. The chromosomes show variation, resulting in the formation of 4 haploid daughter cells, that is called tetrad cells.